Welcome to our second part, that is from 4 Biology Part 2, Genetics. My name is Delvin, Maths and Biology teacher. Last we did up to Mendel's law. We did the first law of Mendel. Therefore, I want us to pick from that. And to proceed, I want us to look at now the crosses, some of the crosses that Mendel did. And before we do the crosses, I want us to look at some of the symbols that are going to be used in the process. Now we need to understand that for us to do the symbols, to represent these symbols, we represent the genes in the chromosome by using the letters. And it's also customary to use a capital letter for the dominant characteristics and small letter for the recessive characteristic. And we also need to know that the gametes are in cycle. And for example, if you have a cross between a tall and a short P plant, whereby we shall let letter T, that is capital T, to represent the gene for tallness, and small t to represent the gene for shortness, the results will be as shown below. That will be the results. The parental phenotypes will have the capital T that is tall, being crossed against the short small t. And the gametes are encircled. So the F1 generation, all of them are tall, as you can see, but they are heterozygous tall. So in simple thing, the fertilization, we are using a checkerboard or a Punnett square, and the F1 generation, the F1 genotypes, all of them are tall, that is capital T small t, and the phenotypic ratio of the F1 generation show that all are tall. Now if you try to, next we do selfing, you self the F1 generation, the outcome from the F1 generation by selfing them, that is the heterozygous tall. If you self them, the cross that is of heterozygous plants from the F1 generation, we have the capital T small t crossed against capital T small t, two parents. The result is as shown where we have one homozygous tall, and then we have two, that is capital T small t, two of them, that is heterozygous tall. Then we have one which is homozygous short. So in the F2 we are having the genotypes, that is tall, capital T, homozygous tall, two heterozygous tall, and one homozygous short. So our F2 phenotypic ratio will be 3 tall and 1 short. That is from that result. Then I want us to proceed and look at something called a test cross. So what is a test cross? Sometimes we always ask ourselves, what's a test cross? Now a test cross is just a cross that is made between the F1 bearing the dominant trait with the homozygous recessive parent. It is also called a backcross because of using the first parent, and it's also called a test cross because it tests the genotype of the individual. Now we have an example shown there. Next, I want us to look at the complete dominance. So what is complete dominance? We know that in this course there's some characteristics. So Mendel happened to choose characters that showed a complete dominance, i.e. he was using the dominant trait that's completely masked, the recessive one in the F1 generation. So that in the F1 generation, majorly the dominant characteristics were dominant. Now in man, certain characters are inherited in the same way. A good example is what we call the color of the skin. The normal color is always dominant to the albinism color. Therefore, we discover that if there's a cross between a normal parent crossing against an albino, we discover that at the end we have the carrier children. So the children will be all normal, but they will have the gene for the albinism, and that is the cross where we have the normal parent against the albino. And the offspring, all of them are normal, but they are carriers. Then we have other characters that show complete dominance in humans, and these are ability to roll the tongue. We have the second one known as polydactyly, and then we have brachydactyly. We have agotroplasia. We'll move to incomplete dominance. Now, incomplete dominance, it's much of the opposite of complete dominance. Now, in this kind of inheritance, there is no dominant or recessive gene, but the two are expressed equally in the offspring, resulting in what we call the blending of the characters. The gene for red color, for example, in cattle, and the gene for white color show incomplete dominance or codominance in some animals, which call them the Frisians. So the offspring are neither red nor white, but are intermediate between the two, and therefore they are said to be roan. In humans, a good example is the sickle cell gene and the normal gene. These are codominant. Therefore, if I give an example, check this one is an example, that is, offspring are neither red nor white, but are in the middle between the two. Therefore, they are said to be roan. Look at inheritance of ABO blood groups in humans. And blood groups in humans are determined by three alleles, A, B, and O. An individual can have only two of these genes. So genes A and B are codominant, while gene O is recessive to A and B, and these are referred to as multiple alleles. So this is an example, that's ABO blood grouping system. We'll have the blood group, the possible genotype, type of antigen on red blood cell, and the type of antibody in the plasma. For example, blood group A, the possible genotypes can be capital A, or capital AO. Why? Because we have said genes A and B are codominant, and therefore gene O is recessive. So in a scenario where we have any other gene, maybe A and O, A will express itself. So the type of antigen on red blood cell will be A, and the antibody will be B. Blood group B, the possible genotypes are capital B, and capital BO, and the type on the red blood cell is B. The type of antibody in the plasma is small m. Then blood group AB, the possible genotype is AB, type of antigen on red blood cell is AB, and type of antigen in plasma is none. We don't have. Then blood group O, the possible genotype is OO, type of antigen on red blood cell is none. Then they have both antibodies A and B. So next we want to look at something else known as the rhesus factor. So apart from the ABO system on the red blood cell of an individual, there is another additional factor known as the rhesus. Now the rhesus factor is responsible for the presence of a protein, antigen D, in the red blood cells, and if blood from a rhesus positive person is transferred into a person without the rhesus factor, we call them the rhesus negative, the recipient's body produces antibodies against the rhesus factor, and therefore this causes agglutination of red blood cells, which can be fatal if a subsequent transfusion with the rhesus positive blood is done. In a case where, for example, the O negative blood O negative may be scarce, and therefore in a case where a transfusion is needed because of emergency, the medical practitioners may wish to use the O positive But if it is done again, if it is a subsequent transfusion in that case, then there will be an agglutination and the patient may die. So next we look at sex determination in humans. In male, two types of sperms are produced, a half of them containing the X chromosome and a half containing the Y chromosome. Now during fertilization, only one sperm fuses with the egg. If it is an X carrying sperm, the female zygote is formed, but if it is a Y carrying sperm, then a male zygote is formed. It follows then that the chances of getting a boy or a girl are a half, what we call 50-50. You need also to note that it is essentially the type of sperm that fertilizes the egg that determines the sex. Therefore, if you cross, that is where we have, that is our cross, we have the female crossed against the male, and therefore XX is crossed against the XY. So if it is the X, because the female have both X's, is the male that will determine, because if it is the X from the male will fuse with the X from the female, and the female zygote will be formed. If it is the Y from the male will be fusing to the X of the female, then there will be a male formed. And therefore, we have equal chances, and therefore there are two female in such a cross against two male. And therefore, the chances are equal. We say they are 2-2. So next, we want to talk about the linkage. Now, the term linkage describes the situation where genes or certain characters are located on the same chromosome. Now, offspring produced by sexual reproduction show only the parental characteristics and only sometimes few new recombinants. Therefore, for example, offspring with combinations of characteristics not found in either of the parents due to crossing over in the first process of meiosis. Now, genes are said to be linked when they are located close together on the same chromosome, such that they are always inherited. Then you have what we call sex-linked genes. Now, these are genes that are located on the sex chromosome. The sex-linkage refers to carrying of the genes on the sex chromosome. Now, a gene for a trait may be present, yet offspring does not show the trait. This happens in humans female, that is XX, where a gene for the trait is recessive. The female acts as a carrier. In humans, sex-linked characters found on the X chromosome include, the first one is hemophilia. Now, what is hemophilia? Hemophilia is a disease that affects the rate of blood clotting, leading to excessive bleeding even from a minor cut. Hemophilia is more common in males than in females. For example, a female may have the gene for hemophilia and not show the trait because the normal gene is dominant over the gene for hemophilia. Such females are referred to as carriers. Now, for example, in this case, we may find that the carrier female of offspring will be carriers, while the other half will be normal. And also, we can discover that half the males will be normal and the other will be hemophilic. A good example is the cross I've done here, where we have two carriers. We have a daughter, all unaffected, but they are carriers. One of them is a carrier, daughters all unaffected, but we have half of the sons unaffected, half of the sons have hemophilia. Next is red-green color blindness. Now, red-green color blindness is where an individual is not able to perceive the red and green colors. Now, it is inherited in the same way as hemophilia, and it affects more males than female. Actually, it is 1 in every 10,000. 1 in every 10,000 males is colorblind, but with less than 1 in 100 million females is affected. It is the inability to distinguish between red and green colors in humans. And this is the cross, that is inability to know. This is the cross. We have an affected female carrier crossed against an affected male. So an affected female is a carrier, and therefore one, as we cross, you'll see that one, that is the one of the son, will be, one of the male will be, male will be colorblind, at the same time will be a carrier. The colorblind at the same time will be a carrier. Now next I want us to do some practice here to know if we are colorblind or not, and therefore I'm going to give you some results for that, and then you can be able to practice. For example, if you have normal color vision, if you have normal color vision, in diagram A you'll be able to perceive 29, diagram B you'll be able to perceive 45, in diagram C you'll not see anything, in diagram D you'll see 26. If you are red-green colorblind, then in diagram A you will see 70, in B you will not see anything, in C you will see number 5, in D you will see nothing. If you have only red color blindness, in A you will see 70, in B you will see nothing, in C you will see 5, in D you will see 6. If you have green color blindness, in A you will see 70, B you will see nothing, C you will see 5, and D you will see 2. Next, I want us to look at genes found on the Y chromosome. Now, these are hair pinna and nose. These are genes which are found on Y chromosome alone, and therefore they affect male only. Number 2 is premature balding, which is also found in male alone. These are genes which they are found only on the Y chromosome. This is an example of those genes that are found on the Y chromosome. Thank you very much, and I wish to stop there. Wish to thank you and appreciate. Thank you very much.